visible? If not,
Yes.
can you see?
No, sir.
Yes, sir.
Okay, okay, just a second. Give me one more second. Okay, tell me if you are able to see.
So
Yes,
now
sir.
it is visible.
Okay, fine. So today we are going to discuss about the intermediary metabolism, how it is being controlled. So it is a continuation of the previous lecture where we have discussed that what is the central theme, what is the central source of energy, how the metabolism starts and how a metabolism is being controlled. So you have basically learned that during the during the cell growth, one of the most important thing is that how cell is concentrating energy from its environment. So first point is that what are the ways in which a cell can extract energy from the source or environment. And second thing is that once it takes the energy from the environment, how it is going to utilize it for various function, for example, synthesize different types of molecules, initially the smaller molecule and then macromolecule. And during this process, you have learned that you have anabolism where the synthesis occurs and catabolism where energy is being produced. So larger molecules are broken down to smaller molecule and smaller molecules are further broken down to the simpler compounds and then you get energy. On the other hand, once you produce this energy is utilized for various purposes inside this cell or inside the body, and then they are going to produce useful molecules. So that combination we are going to talk about today, that how this energy is being produced and how this energy is being utilized. So the whole idea is that the type of reactions which are occurring inside the cell, they are important and we need to know. Further, we have also discussed the concept that whatever the molecule you have, they make a currency of energy. So for example, when we have a produce, we produce, whatever you grow things in the field and then you sell it and you get the currency. Or if you produce something in the market and or you produce some mechanical component and then you make them in such a form, so you sell it and you make money, and this money is a currency you to transact. Same way cell is going to produce different, they are transacting everything and then ultimately they need ATP. Now this ATP is the currency of energy inside the cell. So now question comes that how much ATP should be produced? Should we produce a lot of ATP and keep it as a storage? And what are the feasibility? What are the places where ATP is being utilized? And what are the places where ATP is being consumed? So this concept we are going to talk about. In general, the total quantity of ATP in the body is limited to approximately 100 grams. But on the other hand, you will see that it is utilized in most of the reaction and they are, consumed in a very large quantity. Can you guess how much ATP is being utilized by a, resting human being? If I consider that a human being, a normal human being, consumes almost 40 kg of ATP in 24 hours. And this is a huge amount. So you must be wondering that how this 40 kg ATP is utilized. And, this is not a small amount. There's a huge bulk of amount. So it is important that they should not be stored in the form of ATP. They should be made whenever they are required. So there is a very tight control of how ATP is being produced and how your ATP is being utilized. So for your general, understanding, this ATP is not completely divided into smaller molecule. Rather, ATP is converted into ADP and PI, and then whenever we need energy, whenever we have requirement of energy, quickly it is being synthesized, and for quick synthesis, a phosphoryl group is being attached to ADP, and that's the way the reaction proceeds. We will also talk about how this phosphoryl group is attaching to the ADP molecule. What are the different ways in which it can be done? To give you further idea that it is not only 40 kg, when I'm talking about 40 kg of ATP in 24 hours, it's for resting human being. That means our brain cell keeps working, each cell is a transporting molecule, it is utilizing, it is surviving. So that is where this 40 kg is required. But suppose you are running, you will be requiring, or you are doing some strenuous exercise. What you need is that you need something like 0.5 kg per minute. Uh, and where it is being utilized, it is utilized in the motion, it is utilized in the active transport, it is utilized in signal amplification, and the biosynthesis can occur only if ATP is continuously regenerated from ADP. So that means biosynthesis of molecules, suppose I need protein. How protein is going to be synthesized? Your amino acid is going to form a bond, and this bond formation requires energy. Same way, if I require a polysaccharide for storage, that requires energy. So all those biosynthetic reactions, they require energy, and that is coming from the utilization of ATP. So ATP is continuously being utilized, and ATP is continuously being produced. So we will talk about that kind of exchange of ATP and ADP, which is represented by ATP ADP cycle. So I'll also talk about ATP ADP cycle. So the carbon dioxide is being produced. How it is being produced in the cellular respiration? What happens that carbon is oxidized, energy is used in some cases to create compound with high phosphoryl transfer potential. So what is a high phosphoryl group transfer potential? This high group phosphoryl group transfer potential is nothing but the capability to transfer phosphoryl group from one molecule to another molecule. So can you give me one example of high phosphoryl group transfer potential molecule? The answer is very straightforward. It is a ATP molecule. But if you have seen, I think yesterday we were talking about how much energy is being produced, or how much phosphoryl group transfer potential is with ATP molecule. This can be given by delta G value, which is 7.3 kilocalorie per mole under the standard condition, whereas under cellular condition it is minus 12 kilocalorie per mole. Now, this phosphoryl group transfer potential is present not only with ATP, with other molecules as well. And yesterday we have talked about a couple of such molecules which has a higher phosphoryl group transfer potential than ATP molecule. And if we have such molecules, they can transfer their phosphoryl group to ADP and can synthesize ATP molecule. So, whenever we talk about the ATP synthesis, here you learn that ATP can be synthesized in two ways. One of them is that aerobic organism, what it does is that it has a ultimate electron acceptor. For example, in most of the aerobic organism, you have oxygen as a terminal acceptor of electron. So the phosphoryl group, which is being having high phosphoryl group transfer, is being synthesized by a reaction which is being created by aerobic organism by electron transport mechanism. And this electron transport mechanism is coupled with the oxidation process, oxidation of a carbon dioxide, and that is where CO2 is being produced and ATP is being generated. So it is a cellular respiration. Much more detail we will talk about in the next few slide or in next couple of lectures. So the basic concept which you have to learn is that whenever a molecule or fuel molecule is being oxidized, fuel molecule will be like fat molecule, like glucose molecule or any polysaccharide. When they are oxidized, during the process of oxidation, this oxidation, what will happen? The carbon present in these molecules, they will be converted into carbon dioxide because they will be reacting with the oxygen. So during this process, your carbon dioxide is produced on one hand, but that is not the goal. The goal of oxidation is to produce energy. And slowly you will learn that during the process of electron transport, and what will happen that one of the electrons is being moving to the molecular oxygen, whereas the proton part is pumped out of the mitochondria, or for example from the cell, bacterial cell, and during this process a ion gradient is created, and this ion gradient is responsible for entry, and this is the electrochemical potential, the electrical potential, which is responsible for the regeneration of ATP molecule. So this is a complex process. I'm just giving you some food for thought, and we will discuss it in greater detail whenever we talk about biological oxidation. At this point, it is essential to understand that in aerobic organism, ATP is synthesized during the process of electron transport and oxidation. And this oxidation is the oxidation of your fuel molecule. Then second thing could be uh, during the oxidation process, it is not necessary that always the electron goes to molecular oxygen. Sometimes it will create high phosphoryl transfer potential molecule. And this high phosphoryl group transfer potential molecule will be later on transferring its phosphoryl group to ADP if they are having more delta G of the reaction. So I'll take one example and explain it. For example, in glycolysis, you will learn that the phosphoryl group which is being transferred from phosphoenol pyruvate, that is the one which will be responsible for production of ATP. So you have two processes of ATP generation. One we will call as a substrate level phosphorylation, and there is a second where it is an oxidative phosphorylation. So two types of phosphorylation we'll discuss at the later stage when we talk about the metabolism. Here we are just introducing the concept. Okay, this is giving you that the basic theme that how ATP is converted into ADP. That means your the energy requiring processes like motion. When your muscle is moving, your proteins are changing one shape to another shape, then you require energy. You require it for active transport that you have learned in the classes when we were talking about the transport process, active transport process, where where the molecules they are moving against the concentration gradient, and that's where you require energy. Then today we have discussed about the biosynthetic process where you have to synthesize, whether it is a protein synthesis or it is a polysaccharide synthesis, you require this kind of energy which is coming from ATP. Then you have a signal amplification. This part I have not discussed. This part we'll discuss at the later stage in our course. And this is about how signal can be amplified. You give a small signal and this is getting amplified. That means everything has to be in a larger scale. That means even if we have very small quantity of a molecule, if say in microgram or even lesser than that, they are able to send the message inside the cell. So during this process, there is a signal amplification which is happening, and that is what requires energy. On the other hand, as I said, there are two processes. There are two major way in which ATP is synthesized. This was discussed yesterday. One is a phototrophs, that means photosynthetic bacteria or photosynthetic organism like plants. They synthesize the molecule, and during this process, ATP is being synthesized, or ATP generation molecules are being synthesized. And second way is that oxidation of fuel molecule could be glucose molecule, could be fat molecule. And then today we will talk about, is there any general theme? Is there any general way in which all the molecules are being uh, processed, or all the molecules are producing energy, or they are moving in a different direction? So all those concepts we will be talking about today. Now, in the previous slide, when I say that high phosphoryl group transfer potential molecule, so this is one example where 1,3-bis phosphoglycerate, it is a compound which is synthesized by metabolism of your glucose molecule, and this molecule is converted into 3-phosphoglyceric acid. And during this process, what is happening that one of the phosphoryl group from 1,3-bis phosphoglycerate is transferred to ADP molecule. So this kind of a synthesis is referred as a, what do we call them? What it is doing is that phosphoryl group transfer potential of a compound is coupled with carbon oxidation to ATP synthesis. So what you are looking at is that here, basically your phosphoryl group is a transfer to ADP molecule, and it is happening from the molecule which is synthesized in one of the metabolic pathway. So at this point, the only thing which I need you to understand is that there are some high energy rich molecule which can quickly produce ATP, and these are called as a substrate level phosphorylation. We will talk about that more when we talk about the glycolytic pathway or glycolysis. Uh, the energy of oxidation, this part I have not covered here. We will talk about this a little in detail when we talk about how mitochondria is producing energy, that means ATP synthesis, by re-entry of a proton in the mitochondria. So that's a different concept. And that is where you learn that ion gradient is very important. So ion gradient across the membrane provides an important form of cellular energy, and that can be coupled to ATP synthesis. Details we will discuss about the, or when we talk about oxidative phosphorylation. Here we are just giving you concept of oxidative phosphorylation. So that this is the theme by which your energy is being synthesized, or ATP is being synthesized. So what you have is that the electrochemical potential of ion gradient across the membrane produced by oxidation of fuel molecule or by photosynthesis. So if it is happening in a photosynthesis, what we call photophosphorylation. We'll talk about that. When it happens in the in mitochondria, we'll call it as a oxidative phosphorylation. So oxidative phosphorylation of fuel molecule, as well as during photosynthesis. In photosynthesis, source of electron is, or source of energy is, light energy. Whereas in oxidation of fuel molecule, the source of energy is oxidation of compound. So it's a chemical energy. The other one was light energy. In both the processes, the ATP is being synthesized. Detail I'm not discussing, so then don't get confused. Now, in animals, proton gradient generated by oxidation of a carbon fuel accounts for more than 90% of ATP generation. That it becomes this very, very important process by which ATP is being produced. And this process is called oxidative phosphorylation, as I said. But in case of a plant, it is by photophosphorylation. So in animals, it is oxidative phosphorylation. In plants or in phototrophs, it is by photophosphorylation. The ATP hydrolysis can be used to form an gradient of different types and functions. So first thing is that initially an ion gradient is created because during oxidative phosphorylation, electron imbalance is being created, or protons are being pumped out of the mitochondria. That's why the ion gradient is created. And this ion gradient is utilized for ATP synthesis. And once ATP is synthesized, they can be used for different other ion gradient. As you have already learned that in one case you have learned about this. Do you remember? It was sodium potassium gradient. So if sodium ion gradient is created, you can pump different molecule. You can pump glucose molecule in certain cases. In some cases, you can, pump calcium ion from out of the cell. So there are different processes. And you have also learned that if I block the sodium potassium pump, what will happen? The calcium will not be pumped out of the cell. And this became a very important tool to develop a drug which is known as a cardiotonic drug. I'm just reminding you whatever we have discussed in the previous classes. Now, let us move on to a central theme by which all the metabolism occurs, whether it is a fat metabolism, or it is a polysaccharide, or it is a protein. All of them, they are metabolized by a central theme. So there are three stages, which was discovered or which was told by Hans Krebs. Krebs is very famous, who has given the cycle, which is known as citric acid cycle, or it is also known as a Krebs cycle. Some people they call it the TCA cycle because TCA is the first compound which is made in the cycle. So this cycle is the major source of energy. Let's come to that. But whenever he was discovering, he learned that there are three major components, three major, stages. First stage is the one on the top one, stage one. Here, the macromolecule like fat, like polysaccharides, like proteins, degraded in a smaller component. So this is the first stage of the metabolism, and this is a common thing. So when I say fat metabolism, what happens? What are the fats? Which are we metabolizing? So generally, when we consume oil or a fat, its chemical component or its a chemical component is basically triacyl glycerol, or we also call them as a triglyceride. It could be oil or it could be fat, but both of them they are made up of a chemical which is a triacyl glycerol. That means you have a glycerol conjugated with three fatty acid molecule. So when you break the bond, so these are the ester bond which you break and you get fatty acid and glycerol. During this process, you do not generate energy. No energy is being generated. It's a very, it is just broken down to a smaller molecule. It's like chewing your, food inside your mouth. And then you are breaking down into a smaller molecule. Suppose you have a polysaccharide, say starch, which is major component of our food. What we produce glucose. But other than this starch, if you have any other polysaccharide, which is made up of other individual sugar molecule, and if they can be degraded by the enzymes present in our system, you get other sugars. But all of them, they are pulled together during this process, you don't get energy. Same way, when proteins are being metabolized, you from amino acids, and these amino acids they enter in the next stage. Now it's very interesting that whether energy is coming from fat or coming from carbohydrate or coming from protein, all of them they produce a central molecule. So in the second stage, the process is to produce a molecule which is acetyl CoA, and this is a two carbon molecule. It just contains two carbons. So suppose I have a glucose, it will be metabolized to acetyl CoA. Suppose I have amino acid, it will be produced in the form of acetyl CoA, or it will enter into citric acid cycle somewhere in one of the component of citric acid cycle. Or if I have a fat molecule, it will be converted into fat and glycerol. Fat molecule will be further metabolized, and ultimately even fat metabolism will yield acetyl CoA, and glycerol molecule will enter in the same. Glycerol becomes a carbohydrate. So it again enters through the same pathway and produces acetyl CoA. Have you heard that how this glucose is? The glucose forms acetyl CoA first by entering in a pathway known as EMP pathway, Embden-Meyerhoff-Parnas pathway, that is also known as the glycolytic pathway. And by that pathway, glucose is converted into pyruvate. It's a 6-carbon compound converted into 3-carbon compound, and 3-carbon compound will be converted into acetyl CoA. So the central theme is to produce acetyl CoA. Once acetyl CoA is produced, it enters in a cycle which is known as a TCA cycle or citric acid cycle. And this is the stage where the major energy is produced. So do we produce energy in citric acid cycle? Answer is no. Citric acid cycle, when coupled with oxidative phosphorylation, here you produce major energy. So this is a major source of ATP synthesis. Citric acid cycle will produce useful molecule, and that we will discuss today. That those useful molecules, those are the ones which will be having certain potential. And here the potential is that you have a molecule which has electron donating potential. So far we were talking about phosphoryl group transfer potential. Now we are talking about electron donating potential. So we will talk about those molecules. If we produce those molecules, they are the one which are producing major energy, because these are the electron, and these electrons are finally being accepted by the oxygen, and then during this process ATP synthesis happens. Okay, so the third stage is the major stage which you need to understand and which we need to, emphasize. Now, the metabolic pathway contains a recurring motif. That means you have seen there is a common theme, there is a common mechanism, there is a unifying theme which is responsible for all types of metabolism. So let's see how it happens. So metabolism appears intimidating because of the sheer number of reactants and reaction. Yesterday, I have shown you that how many types of reaction can occur. You have a, network of reaction which are responsible for energy generating and energy consuming process. And that is why it is essential that we should have very core theme-based reactions which are important for managing the complexity of the reaction. And these common theme or these unifying themes are, first is that you are going to have a common metabolite. What I mean to say here is that, for example, you can have acetyl CoA, which is a common metabolite which is being produced, whether it is a carbohydrate metabolism, fat metabolism, or it is the protein metabolism. All of them, they centrally produce a common metabolite, a common molecule. And this common molecule is basically entering in a simple pathway. Suppose I give you a condition where fat is producing X molecule, glucose is producing Y molecule, and the proteins are producing Z molecule. So you have the different molecule. Then you will require three different mechanism to metabolize. And if there are three mechanism, again here there are several reactions. So you will have several set of reaction going through different ways by which it is going to energy. That will be very complicated, very energy intensive process. And that's why these central themes have been approached, where you have a common metabolite, common reactions, and the regulatory mechanism that stems from a common evolutionary heritage. What it means is that you have a common theme by which the similar kind of a mechanism occurring, whether it is a bacteria or a human being, to control the regulation. What I mean to say here is that you have lot of energy requirement. So what will happen, that this cycle will become fast, they will be more operative, they will be, activated. So you will have a mechanism where the citric acid cycle will be activated, and then you will produce more energy. Suppose your cell has a lot of energy, lot of energy. In those cases, this cycle will be shut off, and there are other cycles which will be activated. And those cycles, what they will do? They will synthesize protein, they will synthesize polysaccharide, they will synthesize fat. So you have a common theme by which you can, based on this requirement of energy, based on the availability of energy, you can switch the pathway either from synthesis to degradation or degradation to synthesis. And this common mechanism or this common regulatory mechanism is essential for the reactions. Now let us talk about some of the common carriers of the electron transport. So the activated carriers, they exemplify the modular design. It is like making a modular kitchen or a modular, nowadays everything is becoming modular because you make the unit structure, and these unit structures are fitted in such a way that it can make different types of structures. Uh, some of you might have played with Lego. Have you played with the Lego? Hello?
Yes. Yes, sir.
Yeah. So those are the things which are you have a common motive. And then what you do is that you can make different types of shapes. So here what happens, you have a same theme molecule and they can fit in different reactions. So let's talk about some of them. So these are model design where ATP is an activated carrier of phosphoryl group transfer potential, because phosphoryl transfer from ATP is an exergonic process. Now you have activated carrier of electrons for fuel oxidation. And these molecules are, for example, fuel molecule transfer its electron to a special carrier. So suppose I have a glucose or suppose I have a fat, all of them, they will transfer this electron, which is present in the fuel molecule, to common molecule. And which these common molecules, these are pyridine nucleotides or flavine nucleotides. Have you heard of this pyridine or flavine? I have written in the down a reaction which is employing NAD plus, another is FAD. Have you heard of this NAD or FAD? NAD full form is nicotinamide adenine dinucleotide, and FAD is a flavine adenine dinucleotide. So what are these molecules? 3 days back in a lecture, we were talking about vitamins and coenzymes. So whenever we were talking about the coenzyme, you have learned that niacin is converted into coenzyme, which is a nicotinamide adenine dinucleotide. Another coenzyme is FAD, which is coming from flavine. So these are basically coenzyme. Now, whatever the reaction I have shown, this will also require enzyme. So enzyme is present along with enzyme. These coenzymes, they play interesting role. Here I have not written the name of enzyme because I'm just putting a common theme. It can work with a huge number of enzymes. So these are enzymes which will come under the class dehydrogenase. The hydrogenation and dehydrogenation reaction, they are occurring in presence of NAD plus. And what they do is that NAD plus is converted into NADH, FAD is converted into FADH2, and both ways, what they are doing is that they are involved in the oxidation process. So in the oxidation of a substrate, the nicotinamide ring NAD plus accepts hydrogen ion and two electrons which are equivalent to a hydride ion. So what you are producing is you are producing NADH, which is equivalent to hydride ion. Now, this hydride ion is the major source of energy. How? If I have NADH, and you will learn when we talk about oxidative phosphorylation, that NADH has very high electron donating potential. So if you look at the chart, NADH will be the highest one and oxygen is the lowest one, and that's why electron can flow from NADH to molecular oxygen. So at the end, whenever I have talked about the cycle, what you will learn is that in the TCA cycle, this NADH is being produced or FADH2 is being produced. You do not produce ATP in the TCA cycle, but still it is called as a oxidative cycle. It is a cycle which is mainly for energy generation. So energy generation is in the form of these molecules, NADH molecule or FADH molecule. Now what will happen to these molecules? Okay, so on one hand we have talked about the energy generation by high electron potential in the NADH. There is another molecule which is present, which is known as NADP. It is the same structure, same molecular theme, but it has got one phosphoryl group. I will talk about this very interesting concept. When I differentiate between NADP and the previous one, it is NAD. So here I have written NAD plus. In the next case, I have written it as NADP plus. So the difference is one phosphoryl group which is present in NADP. Tell me, what does it indicate? If I have more NADP, what idea it gives you? Or if I have more NAD plus, what idea gives you about the energy status of the cell? Do you get any idea out of it? When I have NADP plus, this is a phosphoryl group. It tells us that cell is rich in energy because phosphoryl group is attached with this. On the other hand, here phosphoryl group is not attached. That means cell is lacking energy. So what you find is that whenever NAD plus is there, most of the time it is responsible for oxidation, and where your fuel molecule will be oxidized. And during this process, NAD will be converted into NADH. On the other hand, whenever I have NADP plus in the cell, it will give signal for the pathway which will go for reductive biosynthesis. That means it will help you in the synthesis of molecules. That means whenever the cell has high energy, it will go for biosynthetic reaction. When it does not have energy, it will synthesize more ATP. And how do they synthesize more ATP? They will break down the fuel molecule, they will produce more NAD plus, and more NAD plus means you will go for, more NADH, because they are extracting hydroxide ion. And once you have more NADH, you will produce more of the energy. Here, this one is giving you a theme or a modular design where an activated carrier of electron is responsible for reductive biosynthesis. Both of them, they are activated carrier of electrons. Here, this gives you, when it is NADP plus, it gives you signal for biosynthetic reaction. Why it is so? Because many times your precursors are more oxidized than the product, and that is why you require this NADPH to carry the electron. So NADPH, the electron donor in most reductive biosynthesis is NADPH, and the reduced form of this is NADP+. So NADPH differs from NADH in two hydroxyl groups of the adenosine moiety. Adenosine is the same one which you have learned during the structure of nucleotides when we are talking about nucleic acid. And then it has esterified phosphate group. NADPH carries electron in the same way like NADH, and NADPH is primarily used for the reductive biosynthesis, whereas NADH is used primarily for the generation of ATP. So that's a major difference by small molecules like phosphoryl group attachment to the structure. Now there is another very interesting acyl group carrier where you have learned about the coenzyme. Have you learned about the coenzyme? A, this we talked again when we were talking about the coenzymes. So this is a coenzyme A. Its major job is to conjugate with acetyl group. So this reaction shows that when acetyl CoA is present, when it is hydrolyzed, you get acetate plus coenzyme A. And during this process, delta G is minus 7.5. That means it does not require, if I have to transfer acyl group or acetyl group to any molecule, presence of acetyl group CoA can help this process. That means it will not require further energy to produce it. So you have seen acetyl CoA is a very important compound for your metabolic process, and this is being synthesized, which carries high energy, and this high energy will be utilized when acetyl group is being transferred. So then coenzyme A is being liberated. So it is having high acetyl potential. That means it transfer acetyl group, and this transfer is exergonic process, and because of that, the process will become spontaneously whenever the enzyme is present. So ATP and acetyl CoA are hydrolyzed slowly. This is a very interesting point. You have learned, do you recall when we hydrolyze ATP, what happens? You have ATP plus water, it gives you ADP plus PI, and delta G is minus 7.3 kilocalorie per mole under a standard condition. And if I consider the cellular condition, it becomes minus 12 kilocalorie per mole. Okay, so looking at this reaction and looking at the ATP hydrolysis, what you expect is that based on thermodynamics, it is a spontaneous process. And what does it mean? It means that if I take acetyl CoA and water, the reaction should occur. Now, question is that if I take ATP molecule, put them in water, will that be hydrolyzed? Any one of you? Thermodynamics says yes. But what happened? Answer is that both the reaction, whether acetyl CoA is to acetate and CoA or ATP to ADP and PI, Both are of them are thermodynamically feasible process, but kinetically they are not feasible. So how they are made kinetically feasible? They are made kinetically feasible with the presence of enzymes. So that is where the enzyme plays an important role, that lowers the activation energy and carries out the process. And this is essential. Why? Because there's maybe some spontaneous hydrolysis which can take even days in the absence of catalyst. So that means it is a kinetically controlled process. So the kinetic stability of these molecules in the absence of specific catalyst is essential for their biological functioning, and because it enables enzyme to control the flow of energy. Now this flow of energy is again very interesting. So these set of carriers, they are the one which are important for the metabolic reaction. You will be finding this in many reactions. Now we have metabolic processes. So one way of control is that you are controlling by controlling the kind of a metabolite you have, the kind of molecules you have. Another way is that all the reaction, they can be controlled by the enzyme, because enzymes are catalysts. They can make many of the thermodynamically non-feasible process feasible, either by coupling the reaction or by making them react faster, or they are helping the equilibrium to approach fast. So that is where the beauty of the enzyme comes, and most of these enzymes, they are controlled. So if you remember, when we started working with the enzymes, when we started discussion on enzymes, we have discussed one major important point. What we have discussed is that enzyme has a high catalytic power. Second thing we have talked about is that enzymes are extremely specific. It could be Rigo specific, or it could be even stereo specific. Then we have also talked about enzymes are regulated process. So these three points, these three properties of enzyme differentiates them from natural chemical catalyst. And that is what is very important, that you can differentiate them from the other catalysts. So there are different ways in which enzymes can be regulated. I think we will discuss this in the next class. But in general, you must know that the enzymes, their amount of enzyme, catalytic activity, or accessibility to the substrate could be major source of controlling them. So I'll discuss this in the next class. So any questions so far? Because I have only few slides, but then time is over. So tell me any question if you have. Okay, second thing is that we were supposed to decide the timing. So what time is a good time for you to have discussion, or you don't want discussion on your paper?
So can we discuss, and then someone can email you like the feasible timings?
Yeah, email me, but I have a couple of meetings today. So you give me two slots, and either you give me before 12 o'clock or after 6 o'clock.
Okay, sir.
Okay, so you mail it to me, and you can even message me. My mobile numbers are given to you in my all correspondence, and then we can fix it. Okay, I would like that most of you should be present so that your queries are being answered. Okay, then if there are now no more question, and you are going to mail me all the details, so I will sign off. Okay, then thank you very much.
Thank you, sir.